My name is Grace Ayola. The aim of this project is to express my gene of interest, CGB7, into a mammalian cell, CHO cell line, using PCI-NO vector to further amplify and determine the effect of CGB7 on CHO cell line. CHO cell line is gotten from Chinese hamster ovary. PCI-NO has a promoter called cytomegalovirus. Functions of CGB7. CGB7 is a protein coding gene, a subunit of the human chorionic gonadotropin. Its expression has been associated to ectopic pregnancy, and the chorionic gonadotropin stimulates the ovary to produce the steroids that is important for the maintenance of pregnancy. To demonstrate this project, I'll be using the following strategy. I isolate my gene of interest, which is CGB7. I select the appropriate vector, and I use restriction enzymes to cut the DNA fragment and the plasmid, and I incorporate the gene of interest into the PCI-NO vector. Then I transfect it into a mammalian cell. Transformation and selection of vector using antibiotics. The recombinant DNA is placed into the mammalian cell through the process of transfection. I will be using stable transfection. Stable transfection has high level of expression, and the introduced DNA is passed to progenies. PCI-NO has neomycin phosphotransferase gene, which is a selective marker for mammalian cells. The transfected cells will be placed in a medium and antibiotics will be added. Non-growing cells are very resistant to the effect of G418 antibiotics. The transfected cells that carry the plasmid will grow and form colonies, which validates successful insertion. To ensure I generated the correct vector, this is the validation strategy I'll be using. Gel electrophoresis and restriction enzyme. Restriction enzymes is an easy method for screening colonies, and these are the process that will be carried out: isolation of DNA plasmid and digestion. After digestion, we run it on agarose gel electrophoresis to verify the vector of expected size. To transform the vector into a mammalian cell, I will be using the process transfection. There are two types of transfection. We have the stable transfection and the transient transfection. The stable transfection passes the introduced DNA to their progenies. The recombinant DNA is integrated into the cell in small proportion and will be expressed sustainably. The attribute of stable transfected cell is that the recombinant DNA becomes part of the genome and therefore replicates. After placing the vector into a mammalian cell, the transfected cell will be grown in a medium containing at low cell density containing antibiotics, G418 antibiotics. After putting the transfected cell into antibiotics. The transfected cell that contain the plasmid will grow and form colonies, which will validate successful insertion.